Hi guys, and welcome to the next video in the series on biotechnology and genetics. This is number 11, and just a little bit of a look at genetics and the quality of life. So how have some applications of biotechnology contributed to improving the quality of particularly human life? Well, we've started to have a little bit of a look at some of the applications of genetics in the last video, and so in this one we want to have a look at a few specific applications and how they are having an impact on the quality of human life. So we're going to look at four of these in particular, one of which I'll gloss over a little quicker than the others because I assume you would already have some understandings of it. So gene therapy is the introduction of genes into an organism or into a human. Cystic fibrosis is one of the diseases that's been specifically targeted for gene therapy. Transgenic organisms, this has specific applications in the production of insulin, which is for the treatment of diabetes and involves the insertion of human gene for insulin production into a bacterium. Vaccines, there's a range of different applications of vaccines that have been developed through biotechnology, from the very first ones, which was the discovery of penicillin, right through to most of the modern vaccines that you'll be familiar with today, and even vaccines that are under ongoing and continuous development, such as flu vaccines. And there's almost a new flu vaccine comes out every year. And finally, in vitro fertilization, and there's some very interesting implications of around in vitro fertilization, and certainly, I guess, more generally, reproductive technologies. Reproductive technologies have certainly helped us to allow couples that may not, under the natural course of circumstances, be able to have children to still be able to enjoy families. But there has been some implications, I guess, that have been associated with in vitro fertilization, and we'll look at that in a little bit more detail in one of the later slides. So let's look at gene therapy first of all. This is where genes from one organism can be cut using certain types of enzymes, which are proteins themselves, and then inserted into the DNA of another organism, and often this other organism is a bacterium. The reason that bacteria are often chosen is they have something called a plasmid, which is a circular piece of DNA. And so in this circular piece of DNA, a small section can be chopped out and a new gene can be inserted in. So usually that's just, we show that in a slightly different color just to indicate that it's not part of the original. The thing with bacteria is it only takes them about 20 minutes to reproduce. So that rapid reproduction means that we get lots of production of that cloned or copied gene, and we can produce numbers of this very, very quickly. Once we do that, then we can, again, using the same sorts of enzymes that will cut at the same place, remove those genes and then purify them and insert them into faulty cells. Now, one of the techniques, one of the diseases that's being investigated at the moment, not with a lot of success, it must be said, but still early days, is the treatment of cystic fibrosis, which you'd be aware particularly affects lungs, lung tissue, overproduction of mucus, and that can result in a lot of breathing difficulties. And so if we are able to introduce in something like a Ventolin spray, a puffer kind of a spray, for sufferers of CF to be able to breathe in, probably something like a virus which contains the gene. The way a virus operates is to inject its genetic material into the cell, and then the cell will reproduce with the viral DNA in it. But if we put the specific gene that will help to combat the problems associated with cystic fibrosis into the virus, and then the virus injects that into the cells in the lining of the lung, we may actually be able to correct some of those problems associated with cystic fibrosis. As I say, this is still early stage treatment and it's not particularly successful as yet, But this is where all future treatments start, with development, testing, evaluation, change, and ultimately something that is beneficial. Another type of application of technology to the area of genetics is transgenic organisms. As I mentioned before, transgenics is the modification of an organism, usually by the insertion of genes from another species. And as I said, simplest ones to do this with are bacteria, because of their simple DNA structure. And what it means is that we have a gene from one organism which is being incorporated into the genome of another organism. The most, I guess, widespread application of transgenic organisms is the production of human hormones, but it could also include tissues for transplants. Obviously, if we can develop tissues from DNA taken from the host, then the chances of rejection, drop to almost zero. So the problem with transplants is the potential for rejection, but if those tissues were to come from some development of the genes from the recipient, then obviously that significantly reduces problems with rejection. And as I mentioned in an earlier slide, one of the specific applications of transgenic organisms is the production of insulin. And the thing with insulin is we don't even need to inject the gene back into the human. We can actually turn the gene on. We can activate the gene in the bacteria, so the bacterium will produce insulin, which we can then collect and purify, and then that can be used for humans to treat diabetes. Another area is vaccination and gene delivery. I've already talked a little bit through the application of gene therapy for cystic fibrosis sufferers, and at the moment we're looking at the potential for a virus to act as a vector, which is just a transporter, if you like. It's going to move the gene from where it's being produced into where it's needed, which is usually into the lung tissue of a sufferer from cystic fibrosis. And we're going to try and do that in the same sort of way that Ventolin works, by breathing it in, or by using some sort of a nasal spray, in order to help the delivery of that gene down into the mucous membranes and the lining of the lungs. There are other applications, of course, for gene delivery, and they can include plants as well as animals. So, for example, a genetically engineered banana, which is being used to produce a vaccine to combat the hepatitis B virus. So this is using a plant in order to help us develop a vaccine against hepatitis B. Obviously, there's some fairly significant significant diseases around, not just viral diseases, bacterial diseases, fungal diseases, and some of these can be significantly impacting on populations of humans. So where we can develop vaccines to try and combat those, reduce those numbers, and that hepatitis is one, AIDS may be another, I mean, perhaps even some of those really deadly diseases like Ebola. Maybe at some point we will find ways of being able to vaccinate against them, or at least try and reduce the risks and the likelihood of losses of life. Flu vaccines as well. Plenty of areas where there's lots of research and development in order to try and protect people against disease, because prevention obviously is a much more efficient way than trying to deal with treatment once these diseases have gained a foothold. We've also got some development around the embryo, the insertion of healthy genes into an embryo. Now, this is a bit of a controversial one. I will look at, in the final of these particular videos, at genetic screening and its ethical implications. Because of course, one of the things that happens when you know the entire genome, you can identify all sorts of different characteristics of individuals. So testing the embryo, testing its cells, allows us to identify very early certain types of diseases, from general things like Down syndrome, Huntington's disease, Parkinson's disease, all sorts of different things. But to then go that next step and actually insert a healthy gene, you're changing the whole organism, and not just that, you're changing the future descendants. So it's not just that you change the individual, that individual will then pass those genes on to their children and potentially their children's children and so on. So this is actually something which is called germline therapy, because it has implications on the whole of the future line of that individual, not just on the individual themselves. Finally, I just want to quickly have a look at IVF. In vitro fertilization is the process of joining an egg and a sperm outside of the body in a specialized laboratory. The vitro part of IVF is basically referring to glass. So this happens in petri dishes and glass pipettes and those sorts of things. So the egg and the sperm, rather than being fertilized inside of the body of the mother, they are actually joined outside of the body. That's allowed to develop usually until about day 5, because that's the point where the embryo has got its highest chance of being implanting into the walls of the uterus, and it must be protected. So the fertilized egg will just start to go through the processes of cell division through the mitotic divisions, which is what we've said is one of the cell division processes that leads to growth. And so to get from one egg, from one cell to 2 to 4 to 8 to 16 and so on, we need all these mitotic divisions occurring over and over again, copying all that information precisely. So while this is happening outside of the mother's body, it must be protected. And we allow a couple of days, or at least the IVF Australia process allows a couple of days prior to implanting in the mother's uterus. Usually the process of fertilization takes a couple of hours, and it may be more than one embryo which forms. And so if there are multiple embryos, often what happens is they get frozen, and they can be used in subsequent treatments to implant back into the mother's womb or uterus. And you can see, as I mentioned before, it's a blastocyst is a ball of cells, and that's usually around about day 5. So the cells have multiplied and multiplied and multiplied. They're now a big ball of cells. They haven't become specialized into different cells with certain types of functions, although that's been already preempted from their locations, generally speaking. But that's the point where embryo transfer occurs, where the embryo is now going to be transferred into the uterus of the mother, and hopefully at day 5 is the point where the embryo has its greatest chance of being implanted into the womb of the mother. So these are just some of the areas of application of genetic technology and ways in which we've looked at trying to improve the quality of human life by enabling reproductive technologies for parents who were otherwise unable to have children, by helping to detect, treat, and possibly even prevent certain types of diseases, and also to improve the quality of life through quality foods. Thanks for watching.